All right, guys. There we go. Got some interesting settings coming up here. So here we go. So I'm retrying all of this, and we're going to do a complete review of the urinary system, and then I'll post another video that is a complete review of acid-base electrolyte balance. So this one should be kind of short because most of this urinary system side is pretty darn straightforward. So I'm going to make sure we get it better. Okay, so I'm just again going over this sheet that I sent you guys, and I'm going to kind of point out the things that I don't want you to pay attention to, the things I do, as most of you notice. There's some human stuff in here because I used to teach human anatomy and physiology, and thus there's some of the issues there. So I'll tell you what to know and what not to know, basically. So basically, with the urinary system, are going to be your functions, which there are multiple. You would think it's just getting rid of waste, but it actually has a lot of other functions as well, including so it regulates blood volume, blood pressure by controlling how much water it reabsorbs or gets rid of, and the also then thus maintaining concentrations of electrolytes because especially sodium, potassium, chloride, because it can control how much of that it excretes or how much it reabsorbs. And hormones play a role in that too, as you guys have learned. Blood pH is why we have this whole section on acid base, which is really cool. The kidney plays an important role in that. And of course, our elimination of waste. It does drive metabolic products, urea, creatinine. Also can synthesize calcitriol, which helps with calcium absorption. And let's not forget about erythropoietin, which has an effect on telling the body to make more red blood cells. So there you go. Any anatomy, and of course you guys have had, so I don't really do much questions on anatomy. It's going to be more about function. What do these structures do instead of where they are? So there's some human things in here because in the animals it's the right that's a little higher, more cranial than the left, but again I don't care that much about that. So here's just the functions again. So blood volume, composition, pH, electrolyte balance, erythropoietin. So the kidneys play a role when they sense low blood oxygen levels, and the kidney will release erythropoietin into the bone marrow to make more red blood cells. Blood pressure is not just by controlling blood volume, but then I also have renin, which will activate this renin-angiotensin system, which will increase blood pressure. Calcium, again, by calcitriol, activated vitamin D, and that'll tell the intestines to absorb more calcium. And of course, our metabolic waste. So protective coverings we don't really care about. It does have a capsule. Anatomy of the kidney, again, you guys can look at all that if you want to review. We're not concerned with that here. That was, it kind of went into a stopped there. I hope that the recording didn't mess up. But it's just going to look very weird for you guys, huh? Okay, so blood supply, a little bit about blood supply is that you have the renal artery that comes in, and the kidneys receive 20% of cardiac output. That's really important. That is a large volume of blood, right? When you think about your whole body and how much blood leaves your heart, 20% of that just goes to your two kidneys. It just shows you how important they actually are. And then not so much of all the branching, but when you get down to the glomerulus, you're going to get into your afferent arterioles, which brings the blood in, and then your glomerulus, so your glomerulus, your capillaries, and then that goes to your efferent arteriole, which is after the glomerulus, that goes into your peritubular capillaries. The nephron is the structural and functional unit of the kidney. There are over 1 million of those in the kidneys, and that main structure here is the corpuscle, which is the glomerulus, and the Bowman's capsule. So let's get some drawings going on here for you guys. Okay, because the nephron is really important. It's the functional unit of the kidney. So this is stuff that you're really going to have to know. So we're going to go glomerulus. So we'll go afferent, efferent. Okay, so I'm just going to draw it all out here, guys. You guys get all of it? Yeah, all right, cool. So glomerulus, so afferent arteriole comes in, brings in the blood, and goes into the glomerulus. At the glomerulus, that blood is filtered. There's various components of that, fenestrations in the capillaries, pericytes, and things like that that control how much gets through the but basically it's a size thing. So most everything gets through except for when you get to protein size, and anything protein or bigger is not going to get through. Some small proteins can get through. We don't get into the details of that. So that goes from glomerulus, that goes into your tubule. So at the start here is going to be proximal convoluted tubule. I did not draw it convoluted, obviously, but it is very, very convoluted. So it's kind of just all swirling and circling around up in the cortex. So that's the first section, and then I will go into, let's just talk about the sections, then we'll go into what happens in each one. It goes down from proximal convoluted tubule down into your loop of Henle, because it loops around. So this goes, will dive down into your medulla, and then back up into your cortex. And then from your loop of Henle to your distal convoluted tubule, and then that goes down into your conducting duct, which is, then all comes together. Major, minor calyxes, and then into your renal pelvis, into your ureter. So, yeah. This is, I'm assuming we're still going here. Green light is still on. Okay, so we got all of those. So the three functions of the nephron. So remember we talked about functions of the kidney, but now there's functions of the nephron itself. So three things it does. One, filtration. We talked about filtration already. Is blood getting filtered at the glomerulus. Then you have tubular reabsorption and tubular secretion. So most of the reabsorption and secretion happen in your proximal convoluted tubule. And so those three things. We have stuff here about the juxtaglomerular apparatus. I'm not going to require you guys to know this. If you're curious, it's just this group of cells that exist here that are the ones responsible for, like, say, sensing the blood oxygen content and are responsible for releasing erythropoietin. Also, the renin as well. So we won't, I won't concentrate on that at all, and I don't have any questions on that. So let's talk about the cortical versus juxtamedullary nephrons. So there are some numbers here. They're going to be a little different for animals. Actually, they're different for all kinds of animals, which I'll tell you why right now. So you can have cortical nephrons, you can have juxtamedullary nephrons. So cortical nephrons are going to be ones that have the loop of Henle is just a lot shorter, and it barely goes into the medulla at all. Maybe it'll be like right here. Okay, and juxtamedullary ones are going to be these really, really long ones that go deep, deep into the medulla. Okay, and for reasons that I'm not fully going to get into, this is the loop of Henle is where your concentrating ability of your kidney comes from. So how concentrated your urine can make. So the deeper it goes into the medulla, the more concentrated urine can be. And that's just because of different concentration gradients of solute outside in the interstitium. It's called like countercurrent multiplier, which we're not getting into because it actually gets really complex and confusing. So the thing is, though, if this loop of Henle is responsible for concentrating the urine, then you would think if you have more of these guys than you do of these guys, you can produce a more concentrated urine. So these numbers here, for humans, 80% are cortical, 20% are juxtamedullary. Think about cats, or think about any kind of desert animal. So these desert animals do not have a lot of access to water, so they have the ability to make a very concentrated urine. And that's because they have a ton of these guys. And so depending on your animal, it's going to depend on how many of these more percentages you have. I don't even know what the percentages are. Just know relatively, the more juxtamedullary you have, the more concentrated urine you can have. Because if you guys have seen and noticed ever with cats typically have more concentrated urine than dogs. And that's because cats have more of these juxtamedullary ones. That's about how much into it as we can get. We need to get blood supply. We talked about innervation. Not much of innervation going on there. Not much we need to talk about. A couple of disorders I have on here are nephritis, glomerulonephritis, and these are just going to involve, usually due to infection. Glomerulonephritis is something we see more often in animals, and that's going to be these capillaries in the glomerulus that are inflamed, can be caused by usually bacterial infection, often ascending infection from a bladder infection will come and cause the glomerulonephritis. And so we treat with antibiotics. Okay, so what we're going to talk about then, the formation of urine, is filtration, reabsorption, secretion. So we talked about filtration a little bit already. That's fairly simple as far as we're concerned. It's all on molecular size. And then anything is going to get through into here, and everything else just goes into your afferent arteriole. And so reabsorption, there's selective reabsorption going on. A lot of that happens in the proximal convoluted tubule, and so things are reabsorbed from here out of the filtrate, because right, fat filters everything but big proteins. So there's of course going to be things there that get through that you want back in the body, like glucose or something, or amino acids. You're like, hey, I need to keep that, guys. And so that's going to be reabsorbed. And secretion, sometimes there's some molecules that are too big. Maybe some of the drug metabolic products that are too big to get through here, but you still want to get rid of it, right? So it ends up in these capillaries here, and what will happen is then it has some transporters that will take it out of the capillaries and secrete it into the proximal convoluted tubule. And so that's just a little bit of check mechanism for things that get through that you don't want to, and things that don't get through that you do want to. And so with filtration, there's a lot going on here in these papers about glomerular filtration rate. And I'm not going to get into the details. This is the very first step of urine formation. So you have all these different kinds of pressures that drive this. So in your capillaries, you're going to have this, what do we got? So you have this pressure inside the blood, inside the capillaries, like a hydrostatic pressure that is due to the water. You also have a colloid pressure, which is due to protein. And so it's all a matter of how much, it's determining how much filtrate actually gets through. And so if you have a higher hydrostatic pressure inside the vessel, then you can have, which would be like, say, a high blood pressure, for example. So that's going to force more stuff out and into the filtrate. If, for example, you have a blocked cat, if there's a stone and it's blocking the urethra, that pressure is going to back up all the way from the bladder to the ureter up into the kidney and cause at your glomerulus an increased hydrostatic pressure in this space. So if I have increased pressure here, then I'm not going to be able to get as much stuff through, and so I'll have a decreased like glomerular filtration pressure or filtration rate. And so that's the biggest deal of this. I'm not going to ask you any numbers at all. It's more of a relative thing with those two examples that I gave you that are important. So talked a little bit about change in blood pressure and blood volume, how that affects GFR. The renin-angiotensin system I don't talk about a lot, especially not with its relation to GFR. A little bit more detail in all of these other, I really, it's more detail than I think you guys need to know. Okay, so the filtrate, what is it made of? What gets filtered? So it's going to be pretty much the same as plasma, except for large proteins. And so water, glucose, amino acids, urea, uric acid, you know, you guys get what we're reading. You can see all that. So the special wastes that are found, again, are urea, which is the most abundant of the organic wastes. And so that is produced in the liver, and the kidney excretes it. And so this is one of those things that we're always measuring in the blood to measure kidney function. It's called blood urea nitrogen, BUN. And so because when that gets high, that means your kidney isn't able to excrete it. Uric acid is another one that's not that important in animals, but if they're birds, for example, it is. If you get birds, and it kind of has what am I trying to say? With gout, and that's more of a human disease. I've never seen it in animals, as far as your body's inability to deal with that uric acid. In birds, that is how they deal with the urea, or not the urea, the nitrogenous waste is with uric acid. Creatinine is the other one that is generated in skeletal muscle, and so that is our other thing we measure in blood to measure kidney function, is creatinine. And so now we're getting into reabsorption. And so the big thing about reabsorption is, of course, there are some things that are filtered that you want back in the bloodstream. And almost all reabsorption happens in the proximal convoluted tubule. So the proximal convoluted tubule is going to reabsorb almost everything. And so even sodium and water, pretty much everything that gets, most of the stuff that gets filtered gets reabsorbed. 70% of sodium, which water follows. So sodium is by active transport, negatively charged. So the positive charge goes, the negative charge follows. And water's like, hey guys, me too. And water follows too. And that's the main way water gets reabsorbed. So most of that is all in the proximal tubule. Let's talk about glucose. So glucose, 100% 100% of glucose and amino acids are reabsorbed in the proximal convoluted tubule. However, there is a limit of that because there are only so many glucose transporters here that can only transport glucose so fast. And so if I have a ton of glucose coming in, diabetes for example, if I have a ton of glucose coming in, then these can only work so fast, and so some of that glucose is going to escape. And whatever glucose escapes through absorption in the proximal tubule ends up in your urine. And that is the way that then we always test for diabetes, right? That's where the name of it, diabetes mellitus, comes from, sweet tasting urine. Large volumes of sweet tasting urine. That's how they would do it. Imagine, that's how they used to diagnose it, tasting urine. Just think about that. It's awesome. And so that is a threshold for glucose. And I think in cats it's like around 230 maybe. This might be in your book. In dogs it's 180 something. And so then you know if it's spilling over into urine, then you know it's of a certain height, and you don't have to actually measure blood glucose. You can just extrapolate that a little bit from the urine. Pretty cool, huh? So let's talk about ADH a little bit. So antidiuretic hormone. Okay, that's from your hypothalamus. We're not going to get that much into the hypothalamus. The pituitary, that's for endocrine. The next section. And so ADH inhibits the loss of body fluids in times of dehydration. So when you're dehydrated, when dehydration is severe, you're going to have low blood pressure. And this is going to cause you to have a more concentrated urine. When you're dehydrated, your urine is more concentrated because your body is trying to keep hold of all the water that it can. And ADH plays a huge role in that. And so the hypothalamus will sense that, pituitary, posterior pituitary, and then that will release ADH, and the ADH comes here to your collecting duct, which is the other section that's responsible for reabsorbing water, and will add aquaporins. And by adding aquaporins means that the kidney can reabsorb more water. Water goes in, water. And then that'll make your urine more concentrated. That's ADH's big role. So secretion is the process, so that's where substances are taken from the plasma and put back into the capillaries. And so the big thing with proximal tubule are going to be, like I talked about some metabolic waste and things you want to get rid of. And there are other things where secretion happens too, and that's going to be where the kidney is controlling some acid base and some of just electrolyte imbalances, because it has the ability to, say, secrete protons, which we talk about in acid base, and so that's going to secrete protons in there, giving less in the blood. So if you have too many protons, too much acid in the blood, then the kidney can get rid of that acid into the urine. Potassium is the other one. So potassium ions can be secreted into your distal convoluted tubule down over here, and that is going to be under the effect of aldosterone. And then, of course, your penicillin dyes, histamine, creatinine, things are also secreted as well. So, look, aldosterone, our friend aldosterone. So aldosterone is secreted by your adrenal cortex. Again, we cover this in endocrine in more detail. And so if the potassium concentration is too high, then it will cause tubular secretion of potassium and reabsorption of sodium in water. And so that's kind of the effects of aldosterone. And we'll get into the endocrine system, and then we'll talk about additives and the effects of that. Countercurrent exchange, I said I would not talk about. But so proximal tubule is responsible for almost 70% of all reabsorption of everything. And the lipopenile comes down, and this side is permeable to water, but as you come back up into the apennine side, it is not permeable to water. And that is the essence of this countercurrent multiplier, and how we can get the more concentrated urine by that. So urine, 95% water, and then you have mainly urea, uric acid, creatinine, and other electrolytes and things like that. So urine volume, don't worry about it, of course, because that is human numbers. So how is urine different from the filtrate? What things are more concentrated in urine than they are in the filtrate? Urea, creatinine, of course, your waste products, then potassium, phosphate. I don't know how much of these I actually asked, so you don't need to know every single one of those. But potassium is a big one. Urea and creatinine are all big ones that are higher, more concentration in urine than in glomerular filtrate. And so abnormal constituents of urine, so we talked about glucose, it's called glycosuria. Diabetes and Cushing's are going to be the causes of that. And then not really excess intake of sugar or carbohydrates. That you aren't really going to have high levels of blood glucose from that unless you have inherent problems of having diabetes. Protein, so proteinuria, big ones are hypertension. So I talked about if you have more pressure here, it can force more things to be filtered, and a really higher pressure can kind of push proteins through, especially smaller proteins. Also, glomerulonephritis, like the inflammation of these vessels, then that can cause proteins to get through. So seeing proteinuria actually can be a sign of glomerulonephritis. Pyuria, that's what we see are urinary tract infections. So that's going to be it's pus. So it's going to be white blood cells and bacteria. So you all see that on a dipstick very easily. Hematuria, red blood cells, also a sign of urinary tract infection, which is what we see most commonly. But you can have some bleeding occurring somewhere that will also cause that. There's some rare things there. And then any bilirubinuria from liver disease, which you'll also see as jaundice and this together. That's just the kidneys trying to help out getting rid of that excess bilirubin. All right. Elimination of urine so goes from the kidney to the ureters to the urinary bladders, which remember the urinary bladder has a transitional epithelium, so that it goes from those layers when it's empty, they're all bunched up layers, and then as it expands, it's transitional, so it can go from a stratified to a kind of pseudostratified, almost simple epithelium as it gets really large. You have the muscular layers, the detrusor muscles, and you have certain internal sphincters and external sphincters. The neurocontrol micturition, which is the process of producing urine, is quite complex actually. And you have internal, external urinary sphincter, and some under voluntary control. So the process of, say, house training, for example, or even potty training in children, which I'm doing right now with my kids, has to do with learning that control. Of your internal, mostly your internal urinary sphincter, is learning to be able to control that. The urethra transports the urine to the outside of the body, so I think in males that is a much longer path to go around. Male dogs, a lot of male animals have an off penis too, and so as that is traveling around, you can get a stone lodged in there, and that's a very common place as it's coming around. It gets lodged right where the beginning of the off penis is. And of course in the females that urethra is pretty short. Micturition, again, is the process of emptying the bladder. And problems with that we see in animals would be incontinence, so we see that quite often in female dogs. Back when I learned about it, it was still believed to, because I know estrogen will help as a treatment for that, and so it's not as severe. I don't know if more has been done with that. I'm not entirely sure. And that brings us to the end of this packet. I hope I didn't talk too fast, and I'll see you guys again in next week.